So I'd like to introduce our new assistant professor in statistics, Kyle Hasenstab. He comes to us, he was just talking about his circle through the UC schools. He did his BS in quantitative economics and mathematics at UC Irvine. And then he earned his PhD from UCLA in 2015 in the area of functional data analysis. We get him straight from his postdoc at UC San Diego in radiology. His research interests are in statistics and machine learning applications, specifically biomedical engineering. He has expertise in deep neural networks and convolutional neural networks. It was interesting when I was looking on his LinkedIn page, he has quite a wide variety of jobs. He did say he was at the CDC in Atlanta, but he also worked, I think, as a summer intern, probably looked like from the dates at the Census Bureau, and he even was at AT&T Cricket Wireless. So he has a wide variety of expertise and experiences. And today he's going to talk about machine learning techniques for biomedical image characterization. So
Great.
Take it away, Kyle.
All right, thank you. Thanks, Dr. Bailey, for the introduction. And also thank you to Dr. Castillo for inviting me here to talk with you all today at the CSRC. I'll just go ahead and share my screen.
The best approach is to share the slides. Okay, yeah.
Okay, so can everyone see my screen?
I do. So.
Perfect. Okay, so yeah, today I'm going to talk to you about some machine learning techniques for biomedical image characterization. And I apologize for the title. I know it seems like I'm going to do a survey of methodology, but in reality, this is just, I'm just covering a couple of the projects that I've been working on over the past year or two. So this talk comprises two separate topics within biomedical imaging. The first relates to liver imaging. So we're going to assess the diagnostic adequacy of a certain type of liver image called a hepatobiliary phase image within the modality of MRI, or magnetic resonance imaging. The second application relates to the characterization of chronic obstructive pulmonary disease, specifically within the imaging modality of computed tomography, or CT. And in this case, we're just going to relate structure to function, which is a sort of common theme within biomedical imaging. So this is assessing the diagnostic adequacy of these HBP MR images. So I just want to say that this is a highly collaborative effort. I'm working with a team of radiologists over at the Department of Radiology at UC San Diego. I also co-PI'd this project with Dr. Armin Schwartzman, who's a biostatistician professor over at UCSD, and co-mentored one of his biostatistics students, Wen Yilin, who's about to become a candidate, a PhD candidate. So just a bit of background. So hepatocellular carcinoma, it's also known as HCC. It's a type of liver cancer and is a leading cause of cancer-related death worldwide. Now, screening patients at risk for this type of cancer allows for early detection as well as planning for their treatments. So we want to be able to screen patients with hepatitis, non-alcoholic fatty liver disease, as well as possibly fibrosis or even cirrhosis. Because if we were to screen these patients, hopefully every 6 months, we'll be able to detect these cancers when they're very small. Now, contrast-enhanced MRI, specifically using a type of contrast called EVIS. This is an intracellular contrast, meaning the liver uptakes this contrast within its own cells. It provides high sensitivity as well as specificity for the detection of these types of cancers, meaning radiologists are able to see these cancers better. Now, a major problem is the clinical standard for acquiring these images here. They can be quite inefficient, temporally inefficient in terms of the time at which you acquire the image after contrast injection. So an MRI protocol using this type of EVIS contrast for cancer detection, it requires the acquisition of a specific phase called the hepatobiliary phase. So imagine you're a patient, you're laying in the scanner and it's a bit claustrophobic, right? They first image you before they inject contrast, and that's an example image of that is here, where everything's sort of uniformly bright. Then a technologist doing the exam slowly injects contrast, and it goes through your portal vein, your arteries, and eventually into your vessels, and then after a certain amount of time into your background liver here. So for this type of image, this hepatobiliary phase image to have diagnostic value, the vessels within the liver, also used as a proxy for the cancerous lesions, they should appear dark against the background liver, which we call the liver parenchyma. And it's this contrast between the dark vessels and the bright liver that facilitates HCC detection, right? Radiologists see this image and it's easier to spot these lesions. I'll emphasize though that this lesion is fairly obvious. I mean, you can even see it in the non-con image, and I believe this was already treated. That's why there's this mark here. But the goal is to be able to detect lesions when they're very, very small, on the order of millimeters, so that they can, start planning treatments early on. So the time at which this phase occurs, unfortunately, is highly variable, and it ranges from 10 to as much as 60 minutes. So you're talking about a patient laying in this scanner for up to an hour. And the thing is, the clinical standard is to wait about 20 minutes before you just start acquiring these images, right? So already you can see a problem happening. So if the HBP image is acquired too early, this is very bad, because the image then loses its diagnostic value. And hopefully the radiologist catches this and the patient needs to return again for another scan. And this can be costly for the patient as well as the institution. Now, on the other hand, this is less bad, but it's still inefficient. Late acquisitions are inefficient, right? Requiring a patient to stay in the scanner longer than necessary. So why are you keeping them there? So I, along with some of the radiologists over at UC San Diego, we had conceptualized this protocol where images would be acquired at 5-minute intervals. So the patient's in the scanner, you get a pre-con image, then they're injected with contrast, and you start acquiring these images every 5 minutes, along with the standard images that are 30 seconds in. Those are called arterial phase images, but we're focused on this phase here. So once we're imaging them in intervals, we could conceptualize or come up with a sort of a machine learning algorithm that would determine the diagnostic adequacy of each of these images as they are acquired. So the algorithm does need to be fast. Now, the first image that's selected as diagnostically adequate would indicate termination of the exam. So in this case, we're imaging every 5 minutes. This image here is determined to be diagnostically adequate by the algorithm. Therefore, we terminate the exam, and we are able to save 10 minutes from that exam. So early termination of these exams can increase patient comfort, increase patient throughput at the institution, as well as possibly reduce costs, not only to the institution, but hopefully paying that savings forward to the patient. So all of that was just background to just come to our study purpose. So the overall purpose of the study was, could we develop some automated approach, some machine learning approach for evaluating the adequacy of these hepatobiliary phase images for their diagnostic value for eventual screening for cancer. So the gist of it is this: we take this 3D volumetric image or 3D volumetric series of the liver. We propagate it through some sort of algorithm, and this algorithm will produce a binary classification, classifying the image as adequate for diagnostic adequacy, or conversely suboptimal for diagnostic adequacy. So just to define what an adequacy and suboptimality are in the context of our task, an adequate HBP image is defined by vessels that appear darker, also known as hypointense, to the background liver parenchyma. What's nice about this, as I said before, is this contrast between vessels and background liver facilitates HCC detection, because usually when vessels are dark, the cancerous lesions are also dark. Right now, conversely, a suboptimal HBP image is defined by vessels that do not appear darker than the background liver. So this means that if we acquire an image much too early, the vessels might appear brighter because the contrast is still in the vessels, but the liver doesn't have any contrast yet. That's rare if we acquire the images further than 10 minutes. So we tend to encounter this situation here where our vessels are what we call iso-intense to the background liver, where they're just the same intensity because there's contrast in both the vessels and the liver. So we do not want this. So the data was the study was retrospective and cross-sectional, and we had retrospectively collected about 1,201 T1 weighted MR images from 406 patients. Now, as an inclusion criteria, all the patients or all the images were acquired more than 10 minutes after the injection of contrast. So following data collection, we had 2 radiologists independently determine whether or not each of these images were adequate or suboptimal. And these serve as our ground truth classifications. So whenever these 2 radiologists disagreed with each other, then these classifications were adjudicated in a consensus read so that we can have a single classification for radiology. Then, as with any machine learning algorithm, right, you have your training set. It's just a supervised algorithm. So we have our inputs, we have our ground truth outputs. Then we divide them into training and validation data sets. And I want to emphasize that we separate these data sets at the patient level, not the image level, so as to avoid biasing the algorithms that we're going to fit later on. So that's our data. We've collected our data, and now we needed to develop simultaneously 2 separate approaches to accomplish this predictive task of assessing diagnostic adequacy. So the first approach is we call it the traditional machine learning approach. And funny enough, it used to not be called traditional machine learning. It was typically just machine learning, but when these deep neural networks came out, they're just performing so well, and they're such a different class of algorithm that now machine learning algorithms of a certain type are called traditional machine learning algorithms. But we take this traditional machine learning approach where we take our input volumetric image, we then pre-process it in some way, and then extract features from the images. This could be convolving certain filters that you design and so on. Now these features are just numbers describing attributes of the image. And we input these features into a statistical learning algorithm. Here we use a random forest classifier and then evaluate that random forest classifier for predicting the task of diagnostic adequacy using an ROC analysis. Now, alternatively, we use deep convolutional neural networks, which have become very, very popular in the past 10 years due to their great performance on certain competitions. Especially, you'll hear something about like InceptionNet, ResNet. So we take an image, a 3D volumetric image with limited pre-processing, and we propagate that through some convolutional neural network. Specifically, we use the ResNet, the residual network, and then we evaluate that model for performance using an ROC analysis. I'm sorry, I think I hear. Sorry about that. Oh. Okay, so I have these two approaches, right? We have features within approach 1, we have features in approach 2. Then we look at a hybridized approach where we just fuse the features together from its feature extraction step as well as this CNN step. So we literally take these numbers, concatenate them, and then input those into a random forest classifier. So this is the first approach, the traditional machine learning approach. So each liver is just initially segmented using an independently developed 2D liver segmentation CNN. This was developed independently, but in-house at UCSD by a colleague of mine, Kang Wang. And the reason why we segment the liver first is we want to focus all subsequent algorithms just on the intensities within the liver, right? We only want the liver to define what is an adequate image. Then after that, we correct for any background inhomogeneities in the image, and we do this by subtracting a low-order polynomial fit. So basically, you have a B-spline, you fit a B-spline to this image, which estimates a general trend of the image, and then subtract that trend off. Now, the reason why we do this is the patient is sitting in this giant magnet, and this magnetic field is not perfect, okay? So there are inhomogeneities in the magnetic field, which end up as artifact in the images. So you might have bright parts of the liver here and where the magnet's a little stronger, and then maybe parts of the image here where it's not as strong. So this shows up as some artifact, and by detrending it, we make it so that the liver is more homogeneous and that the algorithms, the subsequent algorithms applied, focus more on just the liver instead of that trending. Now, after image preprocessing, we focus on 3 sets of what we call handcrafted features, which we extract from 3 separate methods. Okay, so the first is a very straightforward approach. We just apply a Gaussian mixture model. And the reason why we apply this is we want to capture the separation between dark vessels and bright background liver parenchyma. So that's the Gaussian mixture model FID. This was actually Dr. Schwartzman's idea, where we use Euler characteristic curves to capture the topological structure of the images. And I'll go into what I mean by that in a few slides. And then something more common to biomedical image analysis is texture analysis. So we use something called the gray level co-occurrence matrix, which summarizes sort of homogeneities and inhomogeneities within the texture of the image. You basically extract or compute metrics from this matrix and use those as features for classification in the random forest. So for the Gaussian mixture model, and all we're doing now is just extracting numbers that we feel are appropriate, helpful to predict that task. So we fit a Gaussian mixture model separately to each image. It's just 2 components. And prior to each GMM fit, we do standardize the voxel distributions of each image to mean 0 and standard deviation 1. So this is your standard Gaussian mixture model fit, where we represent the probability density function of the distribution of voxels within the image as a linear combination of 2 Gaussians, right? So our Gaussian parameters are mu1, mu2, as well as standard deviations sigma 1 and sigma 2 respectively, along with our mixture probability pi here. And the way we fit these Gaussian mixtures is standard. It's using expectation maximization, or EM algorithm. This was all done using built-in functionality within scikit-learn in Python. So applying the GMM approach to just a couple images, what we found that the GMM captured pretty large differences in the component distributions of the adequate images. So if you look at the box plot, we have all adequate images on this side and all suboptimal images, with the y-axis showing just the mean difference, mu1 minus mu2, between the 2 groups. And it really does show that there's separability or predictive power in just the means, right? So it's quite impressive how such a simple approach is able to sort of separate the 2 classes a little bit. You could see here how the means are slightly larger in difference than this image here. Although this image is quite difficult because you can see vessels here, but it was ultimately classified as suboptimal by the radiologist due to all this fibrotic tissue. It just makes it a lot more difficult. And just later on down the road, this is why we look into texture analysis as well. Oh, I almost forgot to mention. So from these GMM fits, we only extract 3 numbers. There's mu1, sigma 1 squared, and pi. So we just use 3 variables as input into the random forest from the GMM at this point. So we also use the Euler characteristic curves, and these extract additional information on the topologies of the image. So just to define what an Euler characteristic is for a 3D binary image, it's the number of objects in that image minus the total number of holes in that image. So if you look at the right, here's an example. We have a single connected component object with 3 holes shown in white. So the Euler characteristic is the 1 object minus the 3 holes, which is -2. So in relation to the liver application, we calculated these Euler characteristics across binarized versions of these hepatobiliary face images. And we do this using consecutive thresholds. So basically we have this 3D volume, right? We set a threshold such that all the voxel intensities, all the voxel values above that threshold, we just set to 1. All the voxel values below that threshold, we set to 0, creating this 3D binary mask. Then we calculate the Euler characteristic. Then we move that threshold up, perform the same analysis, and calculate that Euler characteristic. Now, if we keep doing this for several different thresholds, we end up getting this curve down here. And these curves basically describe the topology of the HBP image. Now, in addition to those curves, we also integrated the Euler characteristic curves, and that was primarily just for visualization purposes and to smooth out some of the noise. But notice that there are hundreds of data points here, or the way we organize this curve, there are 100 data points or hundreds of data points. And we typically like to use parsimonious models. We don't want to input a large number of data points into a random forest classifier. Also for computational purposes, it takes a long time to run sometimes. So we refer to dimension reduction techniques to reduce the dimension of these curves down to something a little bit more manageable. So we basically project these functional curves. We consider them stochastic processes and project them down into principal bases that explain the majority of variation within the data. So we're able to capture the majority of variability within these curves using just 4 components, and they explain more than 90% of the variation. Okay. So we end up having 4 scores, just 4 new features, in addition to the GMM features that we use in the random forest classifier. Now, one thing I do regret, I just thought about this the other day, we could have used something like functional partial least squares or canonical correlation analysis, which is more of a guided approach. You project the curves onto a basis guided by the task of assessing diagnostic adequacy. But we're so far into this project, we're probably going to reserve that as a direction for future research as well. So finally, we've extracted how many? About 7 features so far. We're going to look into texture features now. So we used a gray level co-occurrence matrix, which is a method for quantifying texture in the image. So just to formally define it, the gray level co-occurrence matrix is just a matrix of joint probabilities comprising these probabilities Pij. And those represent the probability of 2 neighboring voxels in the image having a certain pair of intensities. So having an intensity I and J. Now, this creates this matrix of probabilities. And then you perform calculations on this matrix in order to extract features that are useful for predictive modeling. So we extract features from this matrix. Now, I won't go into this too much because we didn't dive into the texture analysis too much within this project, but we extract 4 commonly used features from the GLCM. There's energy, correlation, entropy, as well as the inverse difference moment. And the big takeaway here is all of these 4 measures extract in some way the homogeneity or inhomogeneity of the texture within the image. So we have 4 features now that describe texture of the image, along with the Euler characteristic features, which also can describe a level of texture. All right, so we've just collected all of our handcrafted features, and it was quite a bit of work, but now we can extract features from a convolutional neural network. So in addition to handcrafted features, we actually developed a CNN to accomplish this predictive task, this entire CNN pipeline. So we take this 3D volumetric image, propagate it through this 2D liver segmentation CNN slice by slice. And all this does is it segments the liver, and we can populate it with the liver intensities. So all these 2D slices, we just concatenate again at the very end. Then finally, we take the largest 10 slices and then propagate those 10 slices through a 2D 50 convolutional layer residual network. So this is known as ResNet 50. It's a common network that you can just download off GitHub if you wanted to use it. So we train this algorithm to predict a score between 0 and 1. Now, if the score is close to 1, that implies that the image is adequate. If the score is close to 0, then suboptimal. Now, within this network, there are a sequence of operations that are applied to the image, which hierarchically create certain features describing attributes of this image useful for the task. And these features are at the very end of the network. These are the features we want to use. Now, the problem with these networks is the number of features are very, very large at the end. So this particular network had 2,048 features at the end of the network. So what we did was we customized it a bit by appending a fully connected layer of neurons to do an implicit dimension reduction. So we're just essentially projecting those 2,048 features down to 128 features. And the reason why we do this is if we're going to use those features within the random forest classifier, right? These features in the random forest classifier, random forests they don't quite perform well when you have such a sparse feature space that has no correlation with the output. So we wanted to just reduce that problem here, and also for computational issues. So we have those CNN features now. We have all the features we need, and now we can perform some statistical learning. So here we have our 3 sets of handcrafted features, which we just handcrafted by applying these methods. And we have our CNN features, which were automatically learned by the algorithm itself. So we input these for training into the random forest classifier, then separately input the CNN features, and also input them all together by concatenating them just to compare performance. And we compare the performance using an RLC analysis and also evaluate the performance across different training sample sizes, just to really tease out the added benefit of all the work being put into craft these handcrafted features. So this was surprising. This is not shown, but among all the handcrafted features, the Gaussian mixture model features achieved the highest AUC at 85%. And I would consider this somewhat of a complicated task, and we were able to achieve an 85% AUC just using a Gaussian mixture model with 3 numbers. However, there was added benefit to using all 3 sets of features. We increased the AUC to 91%. And some analysis showed that a lot of that was due to the Euler characteristic features. As expected, the CNN achieved the highest AUC at 93%, but we did still see some marginal benefit, some marginal added benefit when we fused the 2 together in a hybridized approach. So we saw that the AUC improved to 94%. But what's really interesting is when we look at the training sample sizes, if we reduce that, which is common in radiology. So we found that the model using the handcrafted features was robust to sample size. So the solid black line here, the AUCs, basically the handcrafted features performed about the same for all these different sample sizes. What's more surprising is it was about 90% AUC or close to it with only 50 examples. This was 50 images from maybe 25 to 30 patients. The CNN wouldn't converge at this point. And for low sample sizes like this, the CNN also performed only slightly better than random. Another thing to point out is when we combine the handcrafted and CNN features, that model consistently outperformed both handcrafted and the CNN only features. However, you could see that they start to converge on each other, and this shows that basically if you have a sufficient number of examples, a very large number of training examples, like 10,000 images, it seems like there would be basically no added benefit of the handcrafted features to the CNN features, possibly because the features by the CNN probably span the same space, or maybe the handcrafted features are in a subspace of the CNN features. So just to conclude this portion of the talk, handcrafted features, although they are difficult to develop, they do not require large sample sizes. I can't say that with great generality, but that is the case in my experience. Now, CNNs are quite easy to implement. Within an afternoon, you could download a network off GitHub. If you have your images organized, you can probably fit one and get going with that afternoon. The problem is they require large sample sizes, and these large sample sizes are difficult to acquire in radiology. First of all, you need the data. And if you can get the data, you need to ask radiologists to sit down and do the tedious task of annotating the images for you to get your training examples. And that can be difficult to do. Handcrafted features, they are more interpretable because we have what we have in mind. We have in mind what we want to extract to accomplish the task. Whereas CNNs, you lose a bit of that interpretability because it's sort of lost in all those computations. However, there are methods around like saliency maps, class activation maps, and activation maximization, which help you identify network attention, but those have their own pitfalls as well. So all in all, CNNs have very strong performance when you have a large sample size, but if you don't have those samples, I suggest approaching the problem using handcrafted features, right? And then you can predict new training examples using this handcrafted feature model. And in a semi-supervised approach, start training your CNN on the predicted ground truths for the handcrafted features model. And then you could go in and sort of correct your ground truths and keep iterating from there. Now, as far as clinical implementation, if we were to implement this algorithm directly in the scanner, which is possible, you can directly implement it within the GE program for GE scanners. The automated algorithm could possibly reduce the examination length for these patients. as well as decrease patient burden and possibly costs. So one thing I didn't touch upon was when I built this ResNet network, I did not use pre-training. So there's something called pre-trained networks, which are networks that are already trained to accomplish certain tasks. You could download their weights, and they were trained on tasks like differentiating dogs from cats, from bridges, and so on. Now, the problem with these networks is they confine your input image dimension to be the same input dimension as when they were originally trained. So this can be a problem in radiological tasks when you have these images where 3D contextual information across slices is required. So what we did in a separate paper, and this was recently published, was I propagated each of the 10 slices independently through the network in a single batch of training. This produced 10 feature vectors describing each image. And note this is trained end to end. So this is all done simultaneously. And then in a feature fusion layer, I just combined those features together. And this way we were able to leverage 3D contextual information as well as leverage the pre-trained weights of the networks. And we saw dramatic improvements in the performance. So we actually found that the AUC improved to 97%, which is quite high. We even externally validated on scans completely outside of the country from different manufacturers, and we're able to achieve a generalization of 95% AUC. So that's pretty much it for the liver talk. Now I can go into characterization of COPD, and this is going to be a little bit shorter of a talk here. Now, this talk or this project was conducted in collaboration with Dr. Albert Shao, who runs the Augmented Imaging Lab over at the Department of Radiology. And I co-mentored a student in biomedical informatics, who's a PhD student at UCSD. So just a bit of background on chronic obstructive pulmonary disease. Now, COPD, it's a progressive lung disease, and it's characterized by poor airflow, right? Obstruction in your airways, and this causes breathing problems. And these breathing problems worsen over time. It's very progressive of an illness. Now, the diagnosis of COPD, it typically relies on measurements from a spirometer. So if you look on the right, we have an individual using a spirometer, and basically you just exhale as hard as you can, and the volume of air you're able to expire from your lungs in a single second is the FEV1. The total volume of air is the FEC, and of particular interest for diagnosing COPD is the ratio between the two. So COPD severity is divided into stages. We call them gold stages, and they're just defined by simple thresholds on these two metrics. So if you have a ratio below 0.7, that means you have some abnormal lung function, you have some form of COPD. It could be severe, it could be mild. Now, given that you have abnormal lung function, abnormal spirometry, you can be further stratified into 4 separate stages, gold 1 through gold 4, with gold 4 being the most severe. So a lot of these patients are really sick. They have a lot of dead lung tissue and emphysema. Now, a very recent study by the COPD Gene Research Group actually showed improvements in the prediction of clinical outcomes, like mortality and progression, when they incorporate imaging into the diagnosis. Remember, they typically only incorporate pulmonary function into the diagnosis. So we need to incorporate imaging into the diagnosis now. So we had hypothesized that information contained within just these images could predict this spirometry-based diagnosis. So why is this important? Well, imaging-based diagnosis can potentially be used as a screening tool for COPD on these large imaging databases that are available in all these hospitals and clinics. So imagine you screen patients throughout this database, and an automated algorithm flags the patient as saying, this person has possible COPD. The flagged patients can then be referred to their primary care provider or their pulmonologist to formally test them using spirometry, taking into account their smoking history and symptomatology, so that they can have a comprehensive diagnosis and plan for treatment. Right? So the overall purpose of this study was, could we do this? Could we relate structure to function? Can we predict these gold stages from these images here? So the way we do this, or the way we conduct this study, is using data from the COPD gene database. It comprises approximately 9,000 inspiratory and 9,000 expiratory CTs. So it's a massive imaging database, along with their clinical data and their spirometric gold stages. Now, just to define what inspiratory and expiratory images are. So a CT acquisition when the patient maximally inhales, that is an inspiratory image. Then when they normally exhale, that is an expiratory image. So we're going to use these two types of images to predict this diagnosis. So a major question we were asking when we started this project was, which features should we extract to accomplish this predictive task, right? There are so many features we can extract. There's a large amount of information in the images. And as a data scientist, the first thing I wanted to do was, well, we can just insert this into a 3D classification network, learn the features, and be able to do this. Now, the problem with that is radiologists have some mistrust. I don't want to generalize that comment, but a lot of radiologists are a bit skeptical about these algorithms because they're not very transparent, right? They're not as interpretable as we want them to be. So in order to sort of come to a conclusion with how we should, extract these features, we decided to take a pathophysiological approach to the problem. Now, what this means is we want to allow our understanding, the clinical understanding of the mechanisms behind COPD to drive the features we extract. We want the biology to drive the features we extract. So what we do is, as an initial attempt, we try extracting only two clinically understood features, right? And the first one's emphysema. Now, emphysema is characterized by essentially dead lung space in your lungs. It's very damaged air sacs within your lungs. And it's a major, major contributor to breathing problems within these patients. So we extract the first feature, which is the percentage of voxels in the lungs below minus 950 Hounsfield units. Essentially, Hounsfield units is a measure of density, which is a unit of measurement of CT. So the reason why this density is so low is because there's just dead space within the lungs. So this is the first feature we extract. Now, the second feature is a bit more complicated than that. It captures the breathing dynamics using both acquisitions. So we first deformably register the expiratory image to the inspiratory image, and this creates a deformed registered expiratory image. Essentially, it's an image that is almost perfectly colocalized, right, aligned with your inspiratory image. Then we subtract the registered expiratory image from the inspiratory image, and this creates something called the lung deformation density difference. It's similar to a parametric response map, if you're familiar with that. We call it this because it's an image subtraction, so we take a difference of the units of measurement, which is density, and we use a deformable registration to do it. So why would this map be useful, right? Well, LD3 values within this map that are close to 0 indicate that air is still trapped within the lungs when you exhale. So imagine you're a patient, you inhale and they image you and you have air in your lungs, so it's low density. Then you exhale, right? And you have poor function, you have obstructed airways, so air is not able to leave, right? So you have air still in your lungs and you end up having low density as well. So your difference between your acquisitions is a small number. Now, the presence of the trapped air within the lungs, we use as a surrogate measure for poor lung function. So we use both of these images to summarize that measure. So the last or second feature we extract is the percentage of voxels in this map below 100 Hounsfield units, of a difference of 100 Hounsfield units. And we determine the 100 empirically in a separate study. So we have our two features. But we have no way of extracting them at this point because they're within the lungs. So in this way, now we refer to some deep learning algorithms to be able to automatically extract those features. And we do this using just segmentation. So we developed a customized 3D UNet CNN to segment not only the lungs, but the lung lobes, each of the 5 lobes, as well as the major airway, or I should say trachea. However, in this study, we only focus on each lung. But we did develop this model for each lobe because eventually we want to spatially characterize the distribution of COPD in the lungs as well. If you want to hear more about this, this was a whole thing on its own. Just feel free to ask me any details after the talk. So we have our segmentation algorithm so we can extract the features. We have our two features which we extract using the segmentation. And now we can predict the COPD diagnosis of spirometry using the simplest statistical learning algorithm, right? It's just logistic regression. So we apply 4 separate logistic regressions using these features as input in order to predict the spirometric-based gold stage diagnosis. So these are the results. So when we use only emphysema, the prediction of spirometry-based diagnosis was actually decently accurate. We predicted gold stage 4 at 93% AUC. However, some of the less severe cases were only performed at 82% AUC, and we really want to focus on these subjects here because these subjects were able to predict ahead of time and start planning for treatment. The gold stage 4 patients, it's a bit difficult because their lungs are so far gone. It's a lot of palliative treatment at that point. Now, when we use air trapping only air trapping and no emphysema, it actually performed a little better. We were able to improve gold 3+ and gold 2+ a little bit. But when the real power came from when we combined the measures and included both of them within these logistic regressions. So we saw an improvement of gold 4 from 0.93 to 0.96. But more importantly, we saw an improvement of gold 1+ from 0.82 to 87% AUC, which is decent performance considering that we only used 2 numbers. Now, in addition to exploration of the performance of these algorithms for predicting just spirometry-based diagnosis, we wanted to see or explore the added benefit of using these metrics, these imaging metrics, along with the spirometry metrics for the prediction of progression. Can we predict whether or not these patients will be worse in 5 years using imaging? So what we found was actually quite interesting. Now, patients with normal spirometry, those patients who breathe normally, they feel fine, but had severe imaging, their imaging indicated otherwise. Those patients had significantly higher odds of progression than those who had normal spirometry and normal imaging. So why is this interesting? Well, patients with normal lung function, probably not even symptoms, just feeling normal lung function, they can still progress. And we might be able to detect this progression using just imaging. So imagine screening patients in a database of ex-smokers and finding out that, hey, you might progress in 5 years, even if you feel fine now, you might want to go on a beta agonist or some sort of inhaler or a treatment regimen. So just to conclude this portion of the talk, prediction of spirometry-based COPD diagnosis, it's plausible using imaging. And imaging might be used to identify potential progression of lung disease in patients that appear to have normal spirometric measurements. As I discussed in the previous slide, imaging can be used to flag patients with possible COPD. And I really like this idea because it really leverages a lot of the databases out there, although this protocol is a little bit different from the standard protocol for imaging. So if we could flag these patients, we can then refer them later on down the road for a comprehensive diagnosis with their pulmonologist. So as far as future directions, we only use 2 numbers. A lot of data scientists might be, and engineers and computer scientists might be shaking their heads like, man, you reduce these 2 images rich with information to 2 numbers and are accomplishing this task. Well, the 2 numbers are highly interpretable, but there is a wealth of data in these images, and we plan on using these entire images for subsequent tasks. And we can even use the deformation field itself, the information on how this lung morphs to the inspiratory phase. Another future direction in terms of statistics is we can look at the probability density functions using kernel density estimation of the voxel distributions within the lungs. And just the shapes of these distributions, the locations, the skewness, certain summary measures of these distributions might be useful in further characterizing COPD. We can also separate these distributions out spatially, right? We have that 3D lung segmentation algorithm which separates out by lobes, and we can see distribution of COPD or further characterize the disease of COPD based on the locations of the lobes. Finally, this is something I'm playing around with a little bit. If you were to take the distribution of voxels within the lungs and maybe do some projection pursuit, find a basis to project onto, and then take those scores and see if they have some predictive power. On the bright side, what's nice about this is you can then refer back to the basis and visualize that basis to see what location of the lungs is contributing most to some outcome. So just to summarize, there's just a ton of data. This is a massive database with images. I'm not a bioinformatician, but there's a large amount of genomic data as well. There's a lot of clinical data, and all of these different representations can be used together in what they call a multi-omic approach, which can be used to characterize subpopulations, right? Predict outcomes like hospitalizations, exacerbations, as well as discover new biomarkers, which can further characterize this, this group of people suffering from COPD. Now, a major problem with the COPD gene database is it's not quite diverse. I believe this was for study design, but it comprises non-Hispanic, white, and African-American research participants. So it would be nice in the future to incorporate a lot of the understudied populations like Asian Americans and Hispanic Americans who typically aren't included in these data sets so that we can characterize those populations as well. Just to acknowledge, this was a collaborative research effort. This is the IDA Lab, who I worked with Albert Shao on the COPD project, along with some collaborators from COPD Gene. Some of you might know Dr. Douglas Conrad. A lot of SDSU faculty work with him. And then also the liver group and the department, or I should say the division of biostatistics. So I'm free to answer any questions, and thanks for letting me talk with you guys today.
So, any questions?
Yeah, can I start?
Absolutely.
Hi Kyle. I really enjoyed it. I've heard parts of it before, and I learn a lot every time I hear new information. In your first example, and you had 128, I guess it was features from the CNN. When you did a random forest and you did variable importance, were all of them important, do you think? Could you have done dimension reduction?
No, that's a great question. So we recently ran that analysis because we did a revision and the reviewer asked the same thing. So we ran variable importance, more focusing on the handcrafted features. And the variable importance of the handcrafted features pointed out it was the mixture probability that was more important, and one of the Euler characteristic features. Now, within the CNN, it was only a few of the features that really popped out. Despite, we didn't enforce any orthogonalization within the network, but a few of those features popped out. And I would say that for low sample sizes, handcrafted features were ranked higher in importance, whereas with high sample size or large sample size, the handcrafted features were ranked much lower importance because the CNN had customized its own features for the task. But I would say only maybe 20 of the features from the CNN seemed really important even. Yeah.
Great, thank you.
Yeah.
Before you ran the random forest with the handcrafted features, did you try any like fully interpretable models, like a logistic regression or something like that, to see how, before you ran the random forest?
We did, but we didn't dive into that in depth. So I believe we started looking into generalized additive models, just looking at some of those functional approaches, but they ultimately didn't perform as well. But the thing is, we didn't do that in a controlled study. That was more experimental at the very beginning before we even had all our handcrafted features. After that, though, we played around with support vector machines, and we found that the support vector machines didn't perform as well as a random forest.
And I think on your results slide, you showed that your hybrid model got a 1% increase in accuracy compared to your CNN. Was that right?
Yes.
Did you happen to do any cross-validation to look at like the variance of that accuracy score?
So we looked at bootstrapped samples within the testing set, but we didn't look at variation in terms of resampling our testing set and training on other samples. So that's a very good point, because I would assume that those two values are probably not significantly different from each other with regards to resampling variation. I would say that definitely the 100 was probably performed significantly worse, but as you get to 200, it's getting closer, and I think we need to explore that a bit more. Yeah, thank you for that. Yeah.
I have a question about your data set. So were there noisy input images, or?
Oh, for which application, the liver application?
The CNN model specifically.
Okay. So in the context of liver application, yeah, a wide variety of images. There's noise attributed to the imaging itself. So
Right.
within MRI, as well as CT, because you have something called low-dose CT, there's just noise attributed to the imaging itself. And the CNN tends to overcome that rather well. But there's also noise within the liver, and that's attributed to liver anatomy. So I didn't really go into this, but a major challenge within liver imaging is you're trying to identify these cancers. And some of the livers are already sick. That's why they have cancer. And those livers are the texture is very rough and bumpy because it's fibrosis. And a radiologist might classify that as adequate to them, but the algorithm might misclassify it because it's seeing uniformity across the liver or homogeneity across the liver. I guess to answer your question, I found that the CNNs could pretty easily overcome that noise related to the actual image acquisition.
Thank you.
Mm-hmm.
Can I go next?
Sure.
Okay.
Okay.
Oh, hi.
Okay, I'm glad I saw your talk today because I missed the one you gave when you did your interview. So that's cool. I have one comment and one question.
Okay.
Let me start with the comment. So I think you know that I do some texture analysis, right?
Yeah.
Okay, so I think we should talk definitively.
Yeah.
The techniques we have, I think they might be very helpful for you. Especially we got something brand new that we are currently writing a paper. So we should talk.
Yeah.
Now my question is about, you mentioned the problem that radiologists, they like to see the interpretability of the features.
Mm-hmm.
I was curious, did you look at what filters the CNN is learning? And can you interpret what those filters are actually extracting?
Oh, wow. Yeah, that's a great question. So there are things called saliency maps. And these saliency maps are essentially, you're taking the derivative of the output with respect to the image, right? So it gives you sort of the network. Now these maps are decent for interpretability. You can see that this is suboptimal. You can't see vessels. There's just general attention around locating the liver. But then here, you could see that it's focused more on the vessels, right? So that's essentially what we can come up with. There are other approaches, but they just don't work as well. This was the most interpretable. The pitfall is there's activation, a lot of activation down here. And this was common across many of the images that I looked at, and I didn't know why. And recently I developed another method, which is still in the planning phases, which simulates a synthetic liver that visualizes the networks of the CNN, or visualizes the features of the CNN.
Okay, cool.
Yeah, so that is definitely a problem.
Yeah, I mean, that's something that most of the time people don't look at. I mean, what does the CNN learn? What kind of filters? But I think that's something that might be valuable to look at. I mean, just to, for ourselves to learn what kind of model the CNN is trying to learn. And maybe we can put some constraints on the learning or on the filters, something like that.
Absolutely. So some networks now, they include orthogonalized constraints on the loss function at the end, just to force them to be orthogonal or maybe even independent if you used mutual information loss. But then in addition to that, that method I was talking about earlier that I'm developing, I found that the reason why this is activated was the most important feature was vessels. The second was texture. Actually, maybe I can show it to you guys now. This is pure access right here. something I'm running right now.
While you're pulling that up, did you look at the saliency maps for the 6% that it wrongly classified?
Salient, yes. So there would be so that was the problem. There would be activation, and I wasn't sure what was activating those voxels. And here's the problem. So that's the problem. We're used to seeing networks where you're classifying dogs versus cats. And then to make sure that the network's focusing on the dog or the being in the picture, you see that it activates around that image. Now, in radiological images, they're not as relatable. You might see activation in a certain area, but what exactly does that even mean? So I found that when I simulate fake livers using adversarial networks, this is the real liver. And this is me toying around with small important feature, big important feature. So I was able to create vessels in the liver. So the most important feature is vessels. It looks at whether or not there's vessels. Then the second most important feature is texture. So it's saying, well, when this feature is large, I get fibrosis, right? And then the third most important feature is general brightness. So when the image is dark here, when the feature is large, it's bright. The liver becomes bright. Now that makes sense because healthier livers tend to take the contrast more, and healthier livers, by taking that contrast, they become brighter than unhealthy livers. And the healthier livers tend to have vessel visibility. So I'm able to, but I'm still tuning this method, but it really shows that like there are other ways that we need to tap into to interpret these methods, for sure. Mm-hmm.
Sorry, Kyle, I'm a bit confused. You just were talking about the health of the liver. I thought that your adequate versus not was just whether the radiologist thought the image was interpretable.
You know, exactly. So whether or not the image was of sufficient quality or for diagnostic adequacy. So the image, the acquisition itself could be fine with no noise, right? But it could be that the liver had such poor liver function that it couldn't take the contrast up fast enough, right? So the vessels were never visible at that point, and the liver never got bright enough to have that contrast between vessels and background liver. So I think you're right. I oversimplified the task earlier in the slides, but separating the two between adequate and suboptimal is kind of, it's a complicated task because not only are you identifying vessel visibility, vessels might be visible, but the liver might be so fibrotic, the radiologist might still render it suboptimal. Mm-hmm. Does that answer your question?
Yeah, it does. I guess I had another question related to scale. Obviously in medical images, you know kind of what a centimeter is, and as opposed to a meter. With most imaging problems which ResNet is used for, you have no idea of scale. So were you able to kind of use that information of scale at all?
Oh, that's a great point. So we didn't. I resized these images without scaling in mind. In other algorithms where that matters a bit more, it depends on the task, I think. So within this predictive task, I believe just the presence of vessels was robust to the scaling of the acquisition, right? The size of the voxels or the slice thicknesses. But there are certain applications where you probably should take that into account, either by standardizing the scaling or giving the algorithm the actual scaling itself. Like you can have an image and then another input showing, oh, the slice thickness for this image was 1 centimeter or something.
And then I guess the final question, just because it's interesting to me, did you rotate the images randomly as you were looking at them? Like, because everything that I guess is you have the patient in the scanner, they're generally going to be orientated in the same way. So was there much of a difference if you randomly rotated the images of the liver?
Yeah, no, that's another great point. So that's actually referring to data augmentation. So in order to make these models a bit more robust to say spatial changes or even noise changes, what I did was I did random rotations, random shifts, even random zooming. just to simulate other sort of in situations or images that we might encounter. And it makes it a bit more robust to those spatial variations. I think I included, it's a while ago, but I infused it with Gaussian noise as well, randomly. And you know how I took the center 10 slices? I would randomly shift by in the slice direction as well.
Okay, thank you very much. Great talk.
Any other questions?
Yeah, I have a question. With the liver, this convolutional neural network, you used the 10 thickest slices.
Yes.
And I was just wondering, because you're trying to find a cancerous growth, wouldn't you want to look for the slices everywhere and not just the 10 thickest slices?
Yeah, no. Okay, so the reason why we selected the 10 thickest slices was we used the visibility of vessels as a proxy for the visibility of cancers. So we actually don't know if any of these liver images have cancers. We just know that if the image itself has vessels visible, then the radiologist can review the image and be able to identify a cancer if it were to exist. Now, the reason why we picked, and this is a good point, because the reason why we picked the middle 10 slices was it's the largest part of the liver, which tends to have all your vessels, the larger vessels. And if those larger vessels are visible, the lesions or the cancers would be visible. Now, another approach would be to take the entire 3D volume and propagate that through and train a 3D convolutional neural network. But unfortunately, 3D networks, they work really well. They work really well, and that's why we used it for the COPD gene data set. But we only had 800 training images, and you could essentially expand your training sample by using a 2D network where multiple slices within a single volume can be considered separate training examples. So that's why we went the 2D approach. We wanted to leverage that sample size.
So it's the cancerous growth. I'm not a doctor. I'm not an expert in this, but so it sounds like cancerous growths usually don't occur on the edges.
Oh, so it's mainly cancerous growths could occur on the edges, absolutely. But we're looking at whether or not the vessels are visible. And if the vessel's visible in the center of the liver, it's likely that vessels are visible or lesions are visible at the edges of the liver as well.
Okay, all right, thanks.
There is a question here in the chat that it says, in order to overcome the lack of liver images, could we generate synthetic data based on the existing labeled data?
Absolutely. So that's been done before, not for this task. And I messed around with it. In fact, going back to this project I'm working on, technically I could use this. That's exactly what I did, where I generated one that's suboptimal and I generated one, these are fake livers, one that's suboptimal, one that is adequate because all the vessels are visible. And I could definitely generate those livers using a generative adversarial network, and then use those as new training examples and essentially expand my training data set to overcome that low sample size. There's an upper bound in terms of how much you can do this, because you might be simulating synthetic data and overfitting your network to synthetic data rather than real data. But there are advantages to doing this, absolutely. It's just a matter if you want to take the time to fit the GANs. They're kind of hard to train, that's the thing.
And did you try to classify cancer versus non-cancer instead of adequate, suboptimal?
No, we did not. And that's a good question, because it's difficult to acquire cancer positive cases. That's actually a harder task. To identify a cancer, it's very location specific. It's not the general overall appearance of the liver. So you might have one here, like a lesion down here. You might have a lesion up here, further down in the liver. So you effectively have a much smaller sample size. So you have to refer to, I would think, other approaches. There are patch-wise approaches where you convolve a patch across the liver, and then just detect whether or not there's a cancer in the patch without looking at the entire liver. But to do that, you would have to find maybe on the order of 2,000 to 3,000 lesions to train on, and that's kind of difficult to find. Yeah.
Okay.
Mm-hmm.
Any other questions? If not, then we're going to thank Kyle for an excellent presentation, very enjoyable and informative. And thank you all of you for attending the Colloquium series, and have a great weekend.